Welcome, friend, to a quiet stretch of time where the lights are soft, the pace is slow, and the story is gentle enough to keep you company as you drift. In this calm space, we will wander through a small, steady corner of microbiology, a place where tiny lives wait in unhurried stillness. Tonight, you and I will explore bacterial persisters, the uninspired state that outlasts antibiotics, and we will do it in a way that is steady, tranquil, and just detailed enough to soothe the mind. If you enjoy winding down with calm science, you can softly tap like and subscribe. And you're warmly invited to let the playlist in the description continue on in the background as you relax. What city country are you watching from right now? Imagine yourself in a dim lab after hours, where the whir of machines is a quiet breath and the glow from instruments is muted and kind. On a bench rests a clear dish with a smooth gel surface, a petri dish like a pale moon held close to your eyes. On its surface, colonies of bacteria form gentle dots, so tiny, so regular, like scattered stars. The air is clean, a little cool, with the faint scent of ethanol that feels like someone tidying the day away. You pull a chair closer, feeling the chair's quiet sturdiness, and you settle in. In this calm, you will picture how some of these tiny cells slip into a kind of waiting, an unhurried posture that stretches time. They are not triumphant, not defiant, not dramatic. They are simply still. The gentle rhythm continues with your breath, in and out, and the story opens like a soft page. You find yourself watching the idea of antibiotics as nothing more than tools that work best on motion, on activity, on the process of living fast. Many antibiotics prefer cells that are busy, building walls, copying DNA, stitching proteins, dividing into two. When all is active, the tools can interrupt these currents and tap the brakes. But in almost every population of bacteria, a few individuals are not busy. They are slow or paused or nearly paused. They carry the same genes as their neighbors, but their inner clocks are set to a sleepy tempo. Antibiotic molecules arrive and pass over them like wind over stones, not because they are stronger, not because they wear armor. They simply wait. The gentle rhythm continues. You can picture a field of wild grasses under a mild sky. Most of the grass sways in the breeze, bright and moving. Hidden among them, a few seeds have already settled in the soil, hushed, not yet sprouting. If a cold front wanders through, the standing blades take the full touch of chill. The waiting seeds do not feel much at all. Bacterial persisters are like those seeds, not a different species of grass, not a new lineage, just a different state, a pause. When conditions ease, they can return to motion, unfurling slowly as if nothing happened. This is not the tale of heroic survival, but a lesson in quiet timing. The difference between resistance and persistence can feel small to the ear, but it is an important, soothing distinction. Resistance is a change in the blueprint, a genetic adjustment that allows growth to continue even in the presence of a drug. Persistence is not about the blueprint. It is about posture. The same blueprint can yield different postures at different moments, in different grills, with different amounts of metabolic warmth. A persistent cell does not grow well, under antibiotic exposure. It mostly does not grow at all. It simply remains viable, like a light turned low, waiting until the drug fades. When the environment opens again, the dimmer can be turned up and the normal tempo returns. This shift is reversible, gentle, and widespread across many species. The gentle rhythm continues. In your mind, you might see a graph, not sharp or busy, but smooth and sleepy. If someone were to add an antibiotic to a flask of cells, the count of living cells would fall at first, then slow, and then settle into a long, gradual tail. That tail represents a tiny fraction that continues to survive for a long time without special tricks, just by being quiet. This pattern is often called biphasic killing, and you can imagine it as a candle that burns bright and then dims to a persistent glow. The flame does not go out quickly because a few stubborn molecules of wax last longer. In a calm lab, a researcher might watch this curve unfold, comfortable in the knowledge that very slow change can reveal deep patterns. How do cells find their way into this slow posture? The answer is not a single switch, so much as a field of gentle nudges and whispers inside the cell. There is noise in life at small scales. The concentration of a protein fluctuates a little from cell to cell. A promoter turns on and off in bursts. A signal molecule is made, then degraded. Imagine a wave tank with tiny random ripples. Most ripples cancel out, but sometimes, rarely, several align, and a larger wave emerges. Not impressive, but noticeable. In a population of cells, these small fluctuations can tilt a few individuals into a low energy state. They make fewer proteins. They stop dividing. They become tolerant of antibiotics that require active processes to do their work. The gentle rhythm continues. Inside the cell, you might picture fitful knots and unknots of pairs of molecules called toxin-antitin systems. The names might sound dramatic, but the reality is quiet. The toxin slows growth, usually by interfering with something essential but reversible, like protein translation. The antitoxin keeps the toxin in check. The balance shifts, sometimes by chance, sometimes by stress. If the toxin is allowed to act a little more, growth softens into stillness. One such toxin is called Hep A, and it can nudge a cell into dormancy-like states by touching the pathways that feed amino acids to the ribosome, the cell's protein-making table. The ribosome itself has gentle ways to rest. There are hibernation factors, small proteins that bind ribosomes and encourage them to sit quiet, as if chairs were stacked neatly in a cafe after closing. You can picture curved chairs on tabletops, shadows layered neatly on the floor, nothing broken, everything ready to restart when morning comes. There is also the language of stress within the cell, spoken by molecules like PEEP-CHEEP, often called the stringent response messenger. When nutrients grow scarce, PEEP rises and tells the cell to slow. Fewer ribosomes are made. Growth is held gently back. This is sensible, like a town dimming streetlights in a long winter night to save energy. In some species, a rise in PEEP increases the chance that a cell will tip into a persister state. You can see how these internal voices create a spectrum of stillness. Not a single on-off switch, but more like a dimmer knob with infinite positions. The gentle rhythm continues. Energy also matters. Many antibiotics need certain forms of energy flow to enter a cell or to carry out their effects. Aminoglycosides, for example, generally need a healthy proton motive force across the cell membrane, a kind of tiny battery, to enter. If that battery is low, the door opens more slowly. A cell with lower ATP, lower energy currency, carries on in a muted way, and some antibiotics cannot bite as easily. Imagine a mailbox slot that opens only when pushed. A slow house has sleepy hands. The mail rests outside a little longer. This is not about cleverness. It is the simplest physics of motion and rest. You may imagine a biofilm now not as something alarming, but as a tranquil city of cells nestled in a jelly-like matrix. The matrix holds water. It holds softness. Nutrients and waste drift slowly through. In the center of the biofilm, the pace is unhurried. Oxygen is sparse. Growth slows naturally. Many antibiotics have difficulty reaching evenly into this environment, and even when they do, their targets may be less active. The biofilm is not a fortress with walls, but a wet meadow where the air is calm. Cells at the meadow's edge feel the winds of change, and deeper in, all is quiet. The gentle rhythm continues. In a focused lab, someone might use a microfluidic device called a mother machine, a chip with narrow channels just wide enough for single bacterial cells. You can imagine these channels as tiny hallways lit by a kind lamp. One cell sits at the end of each hallway, dividing and feeding a line of daughters that wash away down the corridor. Time-lapse images capture their lives in small increments. When an antibiotic flows in, most dividing cells shrink back from the moving front of time and disappear. A few hold still. They do not divide. They become small points of quiet. Hours pass. The antibiotic flows away. The still cells stretch and begin again. Observing this, the researcher does not cheer or despair. They simply breathe and take notes. The story of stillness is told in patient frames. Persisters are not created equal. Some are deeply quiet, others are barely slowed. There's a sense of dormancy depth, a landscape with shallow hollows and deep caves. Shallow hollows allow quick return when the drug leaves. Deep caves require more time and more subtle signals to wake. Some cells may enter what observers call a viable but non-culturable state, where they are alive by many measures but resist forming visible colonies on conventional plates. Yet, if coaxed gently with the right conditions, they can wake quietly, like a faint memory returning. The language of coaxing is simple: a touch of nutrients, a bit of warmth. Sometimes signals called resuscitation promoting factors that tickle the cell wall. The gentle rhythm continues. Consider an ecological view. Silowenwa. In a changing environment, diversity in timing is useful. If every cell commits to growing fast, the population is brittle when a stress appears. If a few hold back, even by accident, the population holds a reservoir of patience. This idea is called bet hedging. Not dramatic, just practical. Seeds sprout in waves, not all at once. Bacteria divide in waves, with some individuals lingering. The ones who linger are not special in their genes. They are special in their moment. In a clinical setting, which we will treat with care and softness, persisters matter because they stretch the time it takes to calm a bacterial population. This stretch can mean longer treatments or a quiet return of growth after a pause. It is not a scene of crisis, but one of patience. Doctors and researchers know this, and their work is careful and methodical. They think about the medicines they choose, not only in terms of strength, but in terms of how they interact with the moving and resting parts of a bacterial world. They consider timing, dosing, combinations that reach active cells and those at rest. They weigh the benefits of allowing the body's own rhythms of healing and immunity to proceed alongside well-chosen antimicrobial support. The gentle rhythm continues. If you like concrete images, imagine measuring persistence on a calm bench. A flask of bacteria grows to a certain density. An antibiotic is added. Samples are taken at set times, diluted, spread on plates, and incubated. Colonies grow where living cells were present, forming little dots. Early time points show a dramatic fall in dots. Later time points fall more slowly. This is the signature. The tail of dots on the plate, dotted like small stars, shows that a fraction remained viable and eventually resumed their quiet division. Many labs have watched this pattern again and again, confirming that this is not an oddity, but a common gentle fact. When thinking about mechanisms again, you can return to the ribosome's quiet posture. During times of stress, some bacteria produce factors called RMF and HPF, ribosome modulation factor and hibernation promotion factor. These partners encourage ribosomes to pair up and rest, like two chairs leaning back to support each other. This resting state is reversible, and it reduces protein synthesis, the everyday hum of building that antibiotics often target. When the environment improves, the factors slip away, and the ribosomes return to their solitary working state, like chairs set back on the floor ready for guests. Proteostasis, the calm management of proteins, also plays a role. When energy is low, chaperones take their time. Proteins fold more leisurely. Some misfolded proteins are sequestered into tiny aggregates, which can act as a sleepy bookmark, reminding the cell of recent stress. When energy returns, chaperones help these proteins refold or degrade, and the bookmark fades. There is subtle evidence that these aggregates can influence the timing of waking in persisters, as if gentle inertia guides the steps back to normal. The gentle rhythm continues. Metabolism in general is a quiet symphony. In persister, cells many pathways idle. The electron transport chain slows. The cell wall turns over less. DNA replication takes a long pause. Antibiotics that attach to moving parts now find fewer handles. Some still act in limited ways, nibbling here and there, but without the regular cadence of growth, their overall effect softens. This is why strategies to address persisters often include luring them back to subtle motion, just enough to expose their vulnerable steps. A small amount of sugar like mannitol or a related metabolite can sometimes gently raise energy levels in sleepy cells and help certain antibiotics enter or act. It is like opening curtains to allow a little morning light in, not a bright flash, just a pale glow that warms the space. Other approaches look for compounds that act regardless of bustle. Some molecules prod the cell's protein degradation machinery in unusual ways. One class, icodepsipeptidase, can activate a protease called SHLP to chew at proteins indiscriminately, including in slow cells. This activity needs careful handling and study, of course, but it demonstrates a principle. If the usual target goes quiet, find a quiet target that remains. In some organisms, medicines that reduce ATP production can press cells into a corner they cannot step out of. In gentle terms, this is a way of closing a door from the inside and quietly turning the lock. The gentle rhythm continues. Phage therapy, the use of bacteriophages that infect bacteria, has also been studied with soft, careful attention. Some phages require active bacterial processes, while others can wait with patience that matches or exceeds that of the bacteria themselves. The interplay here is delicate, a calm dance of two small entities, each with its own tempo, each influenced by the other's state. Researchers also consider enzymes that can gently chip away at biofilm matrices, making the whole environment more open to treatment. Enzymes are like slow rain softening a clay bank, little by little, quietly reshaping the landscape. A beautiful aspect of persister study is the range of time scales. Some shifts into slowness can happen quickly when nutrients fall. Others are the result of prolonged exposure to minor stresses, like mild acidity or waste accumulation. Cells in stationary phase, when the growth medium is nearly spent, are often enriched in persisters. This is not surprising. The room is already dim. The chairs are already stacked. The gentle rhythm continues. If you like to follow threads into mathematics, you can imagine a model where each cell has a chance to switch into a persister state with some small probability and a chance to wake. Also small, with some distribution that might depend on how deep the dormancy is. Add an antibiotic that acts harshly on growing cells and gently on sleepy ones. Solve for the change through time. You would see a fast drop, then a slower, drawn-out phase, a tail. You can vary parameters and watch the weight of that tail grow or shrink. Add noise to the timing of wake-ups, and the curve becomes more textured, but still smooth to the sleepy eye. Watching such curves settle is a quiet pleasure. Persister cells arise in many species, including familiar model organisms. Escherichia coli, with its tidy rod shape, displays them. Staphylococcus aureus, with its tidy clusters, shows them too. Mycobacterium tuberculosis, a slow-growing organism that prefers patience, is often discussed in the context of tolerance and persistence. Yet, in our gentle exploration, we avoid anything alarming. We hold to the idea that slow science, carefully done, with respect and attention, is a kind of lullaby on its own. The gentle rhythm continues. Imagine now a laboratory day devoted to single-cell tracking. A researcher prepares a glass slide, places a thin film of agar, and sprinkles a few bacteria, then covers it with a delicate glass square. Through a microscope, each cell is a tiny dash. Frames are captured every few minutes while the room lights are low. A mild antibiotic is introduced in a slow flow. Most dashes shorten and fade as they cease activity. A small number remain unchanged, slim and quiet. Time drifts. The treatment flows away. The surviving dashes lengthen, divide into 2 dashes, then 4. The researcher notes the timing, the gentle intervals of pause and recovery. The map of life at this scale is beautiful in its simplicity. You might think of the subtle interplay between persistence and resistance. Over long periods, persistence can provide a window of survival that allows rare genetic changes to arise. Not because persisters are trying, they are not trying. They are just present for longer. If a mutation happens that confers resistance, that line can grow once the antibiotics subside. In nature and in human context, both states can coexist. The existence of persisters does not replace the story of resistance. It coaxes you to view the bacterial world through a wider lens. The gentle rhythm continues. Experimental work has explored how many toxin antitoxin systems contribute. In E. coli, there are many such pairs, and they can act in a staggered, redundant way. Some pause protein synthesis, some nick messenger RNA, some stir the translation machinery into temporary slumber. The cell has many ways to slow itself. Each pathway becomes a thread you can trace, and each thread has the same color of calm. None are theatrical. They are folded neatly like spare blankets in a linen closet. An interesting detail fits the quiet mood. The distribution of persisters in a population can depend on the history of that population. If the cells have experienced stress recently, the fraction may be higher. This feels intuitively right, like a room that has already dimmed its lights, being more ready to rest early the next evening. The memory of stress resides in proteins, in metabolites, in epigenetic marks, not elaborate, just enough to nudge probabilities. Those probabilities do not need to be large to matter. Even 1 or 2 cells in a million can shape the tale of survival if time stretches long. The gentle rhythm continues. Testing ideas about persistence involves care in distinguishing it from resistance. If, after treatment, survivors are grown and their sensitivity to the antibiotic remains unchanged, this suggests persistence rather than resistance. If a genetic change is found and survivors withstand the antibiotic while actively growing, this suggests resistance. These are routine checks, not rushed, not hard-edged, but made with soft, steady steps. The biofilm story invites more gentle pictures. Imagine a slow riverbed where layers of sediment have settled over time. The top layer feels the current. Deeper layers feel only seepage and pressure. In a biofilm, bacteria cling to each other and to surfaces embedded in a matrix of polysaccharides, proteins, DNA, and water. The top layers exchange nutrients and gases with the environment. The deeper layers experience scarcity and move less. The community is not uniform. Some cells die and release nutrients. Some cells enter the quiet state. Persisters are common here because quiet is the natural mode in the center. Treating such a community requires patience and often multiple approaches. One may gently degrade the matrix while introducing agents that reach the heart, all while ensuring that the rest of the system, including the person or plant or pipe hosting the biofilm, is not harmed. The gentle rhythm continues. When considering how to reduce the number of persisters, scientists explore sequential therapy. First, wake the cells a little, then apply an antibiotic that requires activity. In some studies, metabolic stimuli like certain sugars temporarily increase the proton motive force, encouraging uptake of particular antibiotics. In others, molecules that disturb the cell's pH handling or redox balance create a moment of vulnerability. There are no miracles here, only gradual improvements. Each small gain can shorten the tail of survival. Each thoughtful step keeps the whole system in mind. You can also appreciate how dosing patterns matter. A single large dose may eliminate active cells quickly, leaving mostly persisters. And the drug may fade before they wake. A prolonged dosing profile or matched combination therapy may keep pressure on as cells slowly return. Pharmacodynamics and pharmacokinetics, the language of how drugs act and move in the body, become part of the lullaby. Peaks and troughs, half-lives and clearance are like a tide schedule. The right tide can smooth the shoreline. The gentle rhythm continues. Some antibiotics act slowly but steadily, even on resting cells, though often to a lesser degree. There are agents that bind to DNA and cause damage regardless of replication, though repairing the damage may itself require activity. There are agents that disrupt membranes in ways that do not fully depend on growth. These avenues are explored with thorough care. The aim is always to balance effectiveness with safety and to understand that the bacterial world operates with myriad small steps rather than sudden leaps. A captivating area of research peaks at single-cell omics. Even in a paused state, cells have patterns of gene expression that can be captured gently and compared. Protein levels can be measured with fluorescent tags. Small RNAs, the quiet regulators of translation, can be mapped. These studies are like listening to a whispering crowd and learning that some whispers are consistent and meaningful. They reveal that persisters often show reduced synthesis of ribosomal components, altered expression of transporters, changes in stress response regulators. Nothing loud, everything subtle. The sleeping ribosome is not alone. DNA can be guarded by proteins that limit access, as if the pages of a book were pinned together to keep them neat in a breeze. Cell wall remodeling enzymes can be downregulated, making the wall's turnover slow. Membrane composition can change slightly, altering fluidity and permeability. Even hydrophobicity can shift. These small touches collectively create a low-profile state. The gentle rhythm continues. When you breathe in, notice that the air is cool and clean. When you breathe out, imagine a bacterial cell exhaling slowly, not literally, but in the sense of releasing effort. This metaphor is imperfect, but comforting. The cell is a chemistry set that knows how to reserve its energy. The tools of antibiotics are also chemistry. Their dance, whether vigorous or slow, is expressive, but not violent. In a sleeping listener's mind, all of this can be a slow dance of molecules drifting and aligning, parting and resting. The measurement of persisters has evolved. Traditional plate counts remain steady and reliable. Newer methods use fluorescence-activated cell sorting to separate cells by metabolic activity, tagging those with low ATP or low respiration. Others use microcalorimetry to detect tiny heat flows, evidence of life at low energy. It is all supremely gentle work based on small signals integrated over time. Watching these instruments trace soft curves on a screen is like watching mist collect on a morning window, the gentle rhythm continues. Around the midpoint of our quiet journey, if your eyes are heavy but still open, you're invited to share a tiny secret. Still awake? Comment the secret word nebula. When imagining potential futures, think of programmable treatments. One might deliver a waking cue for a set period, then switch to a potent antibiotic, or a slow-release system might maintain low-level activity stimulation over many hours, paired with an antibiotic that takes advantage of that window. Such strategies are a kind of choreography. The dancers are small chemicals in small cells. The music is the concentration of metabolites, the pH, the redox potential, the presence of ions. The stage is a biofilm, or a free-living population. The audience is time. Ribosome rescue pathways add another layer. When ribosomes stall on damaged RNA, a system called translation, guided by tmRNA, frees them. In a state of low translation, these rescue events change in frequency. Modulating such systems might influence how long cells sit in the quiet posture. Chaperones like DNA, K, and grow well become a soft background hum, maintaining proteins while the factory rests. Proteases like LAN and CULP tidy up. The cell becomes a library at night with a few librarians moving slowly, restoring order for the next day. You may also picture the gentle roll of efflux pumps, molecular turnstiles in the membrane that move small molecules out. In persister cells, some pumps can be more or less active, and their impact can be significant for particular antibiotics. The interplay between pump activity, permeability, and intracellular drug concentration is complex, but the main idea can remain soft. The cell controls its boundaries with care, letting in what it needs, letting out what it must. The gentle rhythm continues. Stress signals originate from many sources: state of stress, envelope stress, acid stress, hyperosmotic stress, starvation stress. Each signal maps onto regulatory pathways with names that sound like alphabet soup but operate like night watch guards. They dim lights, shut doors, pull shades, patrolling with patience. Sigma factors redirect transcription. Small RNAs tweak translation. Sensor kinases nudge response regulators. The outcome is a lowered hum, a city in quiet hours, not silent, but restful. As time drifts, you are reminded that bacteria live in communities. They speak with molecules that carry messages to neighbors, a process called quorum sensing. In some species, these signals can affect the entrance into and exit from low activity states. In a biofilm, signals also shape the matrix's architecture, encouraging water channels that bring in resources slowly and carry away waste. The result is an organized neighborhood that accommodates both the busier street corners and the sleepy cul-de-sacs. The gentle rhythm continues. Evolutionary thinking offers gentle reassurance. Because environments are unpredictable, strategies that spread risk are often favored not with a decree, but with the quiet arithmetic of survival over many generations. Persistence is one such strategy. It does not need to be perfect. Even a small fraction can be enough to ensure that once a stress passes, some individuals are present to repopulate. The population's memory of stress resides in the ongoing rates of switching into and out of the persister state. These rates can be tuned by mutations that affect regulatory networks. Over time, In a landscape where stress is intermittent, these rates may be adjusted into a useful balance. It is a kind of remembered lullaby that a lineage hums to itself. In thoughtful medicine, practitioners are aware of both resistance and persistence. The two are addressed with complementary approaches. Diagnostic tests can measure susceptibility and guide antibiotic choice. Treatment durations are considered with care, balancing effectiveness, tolerability, and the likelihood that a quiet tale of survival should be trimmed. Adjunct therapies are studied. Non-drug measures, like careful removal of infected medical devices or mechanical debridement of biofilms where appropriate, are considered in a soft, case-by-case way. This is the calm face of complex care. The gentle rhythm continues even within a single presence of an antibiotic. Timing matters. Some antibiotics are time-dependent, where the key is to keep the drug concentration above a minimum suppressive level for as long as possible. Others are concentration-dependent, where brief high peaks deliver stronger effects. In a population with persisters, the time-dependent approach can be helpful when the waking of slow cells is continuous over time. In one mental image, you can see a river kept at a steady height, while small boats stuck in reeds slowly free themselves and float into the current. In another picture, a strong wave knocks loose those already near the surface, but deeper boats remain caught until they are teased upward. The imagery is simple, but it helps the mind rest while absorbing the Peleia. In the calm of night, one can also think about the scale of persister fractions. In many studies, persisters represent a small fraction of the population, perhaps 1 in 1,000, 1 in 100,000, sometimes more, sometimes less. The exact numbers dance, changing with species, growth conditions, and the antibiotic used. The other lesson is about variance. Two flasks that look identical can yield different tales. Noise is not only in cells, but in the world around them. Gentle science accepts this and seeks patterns across many replicates, many days, many slow, careful measures. There are lovely stories of return. After a long exposure and a long wait, the medium is refreshed, and survivors are allowed to regrow. A population that seemed nearly gone returns in pale waves. Colonies appear again on plates. Each colony that appears was founded by one of those waiting cells. The colony is not aware of its founder's patience. It simply grows as any cell would. In this way, persistence is the quiet bridge between tough moments and ordinary life. The gentle rhythm continues. With gentle precision, let us revisit the molecular pieces again. The hippotoxin phosphorylates a component involved in charging tRNA molecules, which are the delivery hands feeding amino acids to the ribosome. By disrupting this path, the ribosome idles, translation slows, and the cell pauses. The antitoxin HIP-B binds HIP-A and keeps it calm under normal conditions. When HIP-B levels fall, the balance tips. Other toxins cleave mRNA or inhibit translation by binding ribosomes. The outcome is the same feeling, a quieting. The cell does not break its machinery, it stores it for later. The stringent response via PPP is coordinated by enzymes like ReLA and SPOST in many bacteria. Starvation for amino acids triggers PIP2 production, which binds RNA polymerase and shifts gene expression toward survival rather than growth. the ribosome's composition of proteins and associated factors changes. The cell's DNA replication may stop at defined points, with enzymes waiting like librarians holding places with small cards. Even motifs like DNA supercoiling adjust slightly, influencing expression in subtle ways. This suite of changes is an exquisite, low-key symphony that keeps the cell alive with minimal expense. We can look briefly and softly at reactive oxygen species, the small molecules that are the byproducts of metabolism and sometimes of antibiotic action. Some antibiotics increase oxidative stress, which can contribute to cell death. In low metabolism states, the production of these species might be lower, changing the balance of damage and repair. Cells in persister states may have more time to manage these species, or they may simply produce fewer of them. This terrain is still being mapped with caution and respect, as measuring such small reactive molecules under gentle conditions is challenging. Bioenergetics can be gently summarized like this. A cell charges its small battery by moving protons across membranes, then allows them to flow back in ways that drive ATP synthesis and transport. The difference across the membrane is like a quiet waterfall. When the waterfall is tall, power is available. When it is shallow, work is limited. Some antibiotics need help from that waterfall to enter. A shallow fall means a slower flow of medicine inward. Waking cues can raise the fall slightly and help the medicine drift over the rocks. The gentle rhythm continues. There is also a connection to cell size and shape. Slower growing cells can be smaller, with less surface area and volume to maintain. Their membranes may be slightly stiffer, their cytoplasm more crowded. Molecules move more slowly. Binding partners are found less quickly. Antibiotics, dependent on active processes, thus find fewer opportunities. If you imagine a boulevard at night with few cars, a traffic camera will capture fewer interactions than at noon. The camera is not broken. The road is just quiet. In the quiet of thought, you may imagine a scientist looking for chemical markers of the persister state. Perhaps there are phospholipids that are enriched, or short peptides that accumulate, or particular carbohydrate residues in the cell wall that change. Each candidate requires careful validation, not for drama, but for truth. If a marker can be found, diagnostics could one day estimate, even roughly, the fraction of persisters in a sample. This could inform treatment timing or combinations. If no universal markers exist, that is also meaningful. It may be that persistence is a posture reached by many pathways, each leaving different scuffs on the floor. Persisters are not immortal. They can be harmed by prolonged stress, by nutrient exhaustion beyond a point, by changes they cannot accommodate. Their patience is long, not infinite. In our calm narrative, this is reassuring because it reminds us that persistence is a part of normal life cycles. The planet's bacterial communities have weathered dry seasons and wet seasons for billions of years by using timing to their advantage. This is part of the quiet wisdom of life. You might now imagine a gentle conversation between two worlds, the human world and the microbial world. Humans created antibiotics in the 20th century, harnessing natural molecules and synthesizing new ones. Bacteria, ancient and adaptable, responded as they always have, with shifts in genes and shifts in states. The meeting point has found a durable rhythm. We adjust therapies, refine stewardship, invest in discovering new agents, and learn to appreciate the difference between fighting and guiding. Bacteria continue their cycles. With each passing year, the science becomes more careful, more exact, more respectful of complexity. The gentle rhythm continues. One might ask how persisters influence the development of chronic microbial communities in places like soil or water. In these vast ecosystems, stressors are mild and varied. Drying and wetting, hot days and cool nights, sudden nutrient pulses from decaying leaves, brief exposures to toxins. A small fraction of cells that linger can carry the community through these cycles. When conditions are kind again, the persisters seed the next wave of growth. This is a pattern repeated in countless microhabitats, from puddles to the pores of rocks. The metaphor is quiet and durable. The genetics of switching rates is a small, elegant area. Mutations in regulators can increase the fraction of persisters. Some strains show a hyper-persistent phenotype, not because they are genetically resistant to drugs, but because their natural probability of entering a slow state is elevated. Studying such strains helps researchers find the nodes in the network that favor quiet. The names of these genes can be long and cryptic, but their effect is simple: more stillness per unit time. The gentle rhythm continues. There is also the question of memory, or hysteresis, in switching. A cell that recently left a persister state might have a predisposition to re-enter if stress returns. This can be due to residual aggregates, modified chromatin-like structures in certain bacteria, or altered metabolite pools. The shapes of these memories can be sketched and measured. Slowly, a map emerges in which entry and exit rates depend not only on current conditions, but also on recent history. The logic is that of a thermostat with a tiny lag, a familiar kind of stability. To see persistence in a different light, step away from antibiotics altogether and think of predation. Protists and other microbes sometimes feed on bacteria. A slow or altered state may reduce visibility to these grazers. Some biofilm organisms respond to grazers by increasing matrix production, which naturally increases internal stillness. Thus, the persister state can serve multiple soft purposes, not designed, but arising from the same foundations. A note about language may be helpful. The phrase uninspired state can sound like a joke about boredom. Here, it is taken seriously as a metaphor for minimalism. The cell in a persister state is doing what humans sometimes do near bedtime. It reduces activity. It cuts out unnecessary tasks. It waits. There is a beauty in this. In a world that rushes, the cell chooses not to rush. In our quiet hour, that choice feels wise. As we approach the last stretch of our calm journey, imagine once more the petri dish under lamplight. The agar surface is clear. The colonies small and round. You lift the lid gently and touch a sterile loop to one colony, streaking faint lines across a fresh plate. You incubate it and In the morning, a soft constellation appears. Some of these dots might be descendants of cells that weathered a long night of antibiotics. By pausing, they carry no new weapons. They carry only timing. You hold the plate, turn it slightly, and see the sheen of the medium under the light like a shallow pond under a moon. Now think of breathing. In this moment, your own body follows cycles of activity and rest. Organs hum at their own pace. Cells repair and replenish during sleep. It is delicate, and it is wise. The relevance of bacterial persistence to your day is not about worry. It is about appreciating that nature uses rest as a strategy at every scale. From galaxies coasting to molecules diffusing, from seeds waiting to cells pausing, rest is part of survival. The gentle rhythm continues. Tranquil futures in research include creative diagnostics that profile metabolic state, therapies that pair gentle wake-up cues with precise agents, and stewardship that treats antibiotics not as hammers, but as instruments in a well-tuned ensemble. Education will likely continue to emphasize the difference between resistance and persistence in clear, approachable ways. People will learn to recognize that finishing a treatment is recommended for reasons that include both community protection and the small reality of persisters that concede a return if left unattended, while future guidance refines that advice with nuance, guided by ongoing trial data and careful professional judgment. It is always a balance between simplicity and precision. In outreach, the message is delivered with kindness, reminding listeners that these decisions are personal, guided by healthcare professionals, and rooted in gentle biology. The gentle rhythm continues as we look again at the architecture of cell walls. In some bacteria, cell wall synthesis is organized at mid-cell during division, a bustle of enzymes handing molecules along like artisans in a quiet workshop. Antibiotics that inhibit the creation of cell wall crosslinks are most effective when the workshop is busy. When the workshop is in a low activity mode, the flow of material along the benches slows, and the antibiotics' entry into the music is delayed. Other antibiotics target DNA curase and topoisomerase, enzymes that manage the winding and unwinding of DNA. If DNA replication is at rest, if transcription is minimal, these targets engage less often. The workshop still exists. It is simply between projects. Ribosome-targeting antibiotics bind to the multi-part machine that stitches proteins by reading mRNA templates. There are many different seats for these antibiotics, some on the small subunit, some on the large. They can misread, stall, or block. When many ribosomes are hibernating, fewer seats are occupied. The roughness that would typically appear in the cell's output is less relevant because the output itself is slow. The gentle rhythm continues. We can revisit the shape of the kill curve once more because repeating images help the mind relax. Imagine a soft slope down from a hill to a valley. At first, the descent is steep. Then it levels out and slowly continues. The deeper valley is reached only after a long walk. If you map the number of bacteria on a logarithmic scale against time of exposure to an antibiotic, the line is not straight. It bends. The early slope reflects the killing of active cells. The later, gentle slope reflects the lingering of persisters. There can even be a triple phase curve with distinct subpopulations of different dormancy depths. These pictures may be drawn for different antibiotics, for different starting conditions, for different species. They share the same geometry. In single-cell studies, one can collect genealogies. A lineage tree grows across the page. Each split represents a division. After a treatment, branches abruptly end, except for a few that continue as faint lines, unbroken. Those are descendants of persisters. In some experiments, the probability of persistence seems to vary with the age of the cell or its position in the division cycle at the time of treatment. Cells just born may behave differently from cells approaching division. The explanatory story remains the same: timing and state. The tree, like a family tree sketched in pencil, is a map of quiet survival, a final scene to carry you forward. You sit at a wooden desk that has a smooth surface, worn by many hands and years of careful work. Your fingers trace the grain, slow arcs that feel grounding. On the desk is a notebook. You write a simple line: persisters wait without haste. You feel that line settle, like a leaf floating down on still water. Close to the notebook sits a small glass bottle with a rubber stopper, the kind used in labs to hold reagents. It is empty tonight, just a reminder of routines that take place in the day. There is comfort in empty tools, at rest. The gentle rhythm continues. The night deepens. You can let the details loosen and drift. The names of molecules need not be held tightly. What stays is the shape of the idea. Within populations of bacteria, some cells sink into the soft chair of low activity. Antibiotics pass. These cells neither resist nor succumb quickly. They persist. When the environment opens, they return to regular life and become ordinary again. This pattern repeats across organisms and conditions shaped by genetics, metabolism, stress responses, and community structures like biofilms. Scientists watch and learn, not with alarm, but with steady attention. You are almost at the end now. The topic has been quiet and rich. If you choose, you can imagine the next video continuing as you fall asleep, moving from persisters to other forms of microbial patience, or perhaps to seeds and spores and the chemistry of rest in many forms. Or you can simply listen to your breath, to the faint sounds in your room, to the friendly silence of a channel that values softness over force. The gentle rhythm continues, and your eyelids may grow heavier. Thank you for spending this serene time exploring bacterial persisters with me. You are safe, you are cared for, and you can sink into comfort now, letting science become a lullaby that hums in the background. If this calm journey helped you rest, you can lightly tap subscribe and leave a like so we can keep releasing videos this often. And if you want the learning to continue in a whisper as you drift, you can let the playlist in the description play on. Sleep well, and may your night be unhurried and kind.